Hi, welcome back. Before we get into meiosis, I wanted to just spend a couple slides taking a look at cell cycle control and cancer, which is the outcome of improper cell cycle control. So when we are taking a look at cell division in this concept of mitosis, not meiosis yet, but with mitosis, when cells are just making a copy of themselves for the purposes of repair or growth or maintenance, we want to make sure that those cells, when they're going through that process of mitosis, are tightly regulated. Because in a multicellular organism, uncontrolled cell growth is a really dangerous thing. So we want to make sure that that's following proper rules and regulations. So in our cells, we have kind of this built-in control system to make sure that cells just don't go through mitosis any old time they want. So here's a picture of the cell cycle that we saw at the beginning of the first kind of introductory mini lesson. So we can see the interphase, which is made up of the G1, the S, and the G2 phases, with a sandwich of the little M phase in there, which we just went through, which was mitosis and cytokinesis. So built into this cell cycle, we have these checkpoints. Now, they're not physical checkpoints. They are enzymatically driven communication, like cell signaling, which we're going to be doing cell signaling at the very end of this term because we rearranged that. That's chapter 6. But you should still be able to understand this even without having that background. So a checkpoint, and we'll see some of the molecules in the next picture, they're just, you have to have a certain amount of a particular molecule to allow the next stage to happen. So there's only 3 checkpoints shown in this diagram. There's quite a few more that are not shown that are a little bit more in detail that we're not gonna, your book just doesn't cover those. So the first one we're gonna take a look at is this number 1 they're calling is the G1S checkpoint. So the G1, remember G1 is our gap phase, our first gap phase. Like I said, it's growth, the cell's doing its thing. But let's say it gets a signal that it needs to divide. It's a skin cell, it needs to, like the stem cell for the skin, that we need to replenish the cells that are lost from the surface. So this first checkpoint, before it's okay to replicate the DNA, it's gonna make sure it has everything it needs. Does it have enough nucleotide raw material to make a whole new copy of all your DNA? Does it have enough nutrients to make the ATP needed to do all that DNA replication? Does it have the proper enzymes to do that? Here comes my cat. And if it doesn't, that checkpoint will not be okay to go. It'll stay in that phase, in the G1 phase, until the requirements are met before it can go into the S phase. So this is the G1S checkpoint. The second one is called the G2M checkpoint. So this is after DNA replication. It's after most of the growth 2 checkpoint, or after most of the growth 2 phase. And this will make sure that there's no severe damage to the DNA. So we've gone through our DNA replication here in the S phase. And before we split and send that DNA off into our daughter cells, there's like a proofread or a double check to make sure that the DNA isn't damaged before being deposited into the new generation of cells. And then the last one they have listed here is this checkpoint 3, which is the metaphase anaphase checkpoint. So remember, metaphase was all the chromosomes, replicated chromosomes are lined up in the middle, and then anaphase are pulled apart. So at this checkpoint, it's to make sure that all of those spindles are properly attached to the kinetochores, because if you don't have them properly attached, what happens is instead of splitting, you might get 2 chromosomes get pulled to one or the other sides, and that's going to make a cell have one too many of a certain chromosome, and the other cell will be missing the chromosome. This is a process that's called non-disjunction that we'll see more problems when we get to meiosis. In the production of like eggs and sperm is a bigger problem than just like a skin or a body cell. So those are some of the checkpoints that your book describes, just to give you an idea that there are these built-in regulations to make sure that when the cell goes through this process of replication, it does it with high fidelity. We do not want mistakes as we progress through this, and we do not want cells to go through cell cycle without control or without regulation. So here is a look at some of those checkpoints. Really, like I said, they're not a physical, like a stop sign or anything like this. They are a collection of enzymes that cycle in concentration based on signals that are being sent throughout the cell. So the two major players here is what we have, what's called cyclin. So that's the purple, right? So that's this guy right here. So the cyclin protein cycles based on what's happening in the cell. So it accumulates, right? So we get high levels of cyclin. And then we have these other proteins called, or they're enzymes, they're called CDKs, which stands for cyclin dependent kinase. Cyclin dependent kinase. Okay, so it's an enzyme. We know it's got the ASE ending. So the CDKs, when they bind, when these cyclin levels kind of reach a peak at these various checkpoints, they'll bind to the CDK, and now you have your CDK cyclin complex. And for this particular step, this was the M, triggers the M phase, of allows the cell to progress from G2 to M. When you have a proper amount of the cyclin and the CDK, that's what will allow the go into mitosis to take place, into the mitosis phase. And then after it's done its job, it will split. The cyclin is degraded, and then it won't, so it cannot go into another round of division until it builds itself up again, or sorry, until the CDK builds itself up. And until we reach another concentration of this CDK and cyclin to allow the cell to do this again. So there's a whole family of different kind of cyclins and CDKs that allow for those different checkpoints to take place. When that gets broken, if something in that cell cycle control is damaged or is unregulated, this is when we can start seeing cancer or tumors as a byproduct of that. So there are some physical signals that normal eukaryotic cells have to prevent them from growing out of control. So the first one is called anchorage dependence. So what that means is if you have a normal eukaryotic cell, it will only be able to replicate when it's stuck to something. It's anchored onto something. So in this case, it's like anchored to the bottom of a petri dish. If you were to take a cell, a normal cell, and just let it float around in solution and not be stuck to anything, it won't replicate. It's not getting the signal that it's stuck to a surface for it to replicate, because in our bodies, we don't have a lot of free-floating stuff. And we have our blood cells, but those are made in bone marrow. They're not made in the fluid environment of the blood. They're made in a solid state, and then once they're mature, they can go and circulate through the blood. And so anchorage dependent is part of these checkpoints to make sure that the cells are not going to grow out of control. Another regulation was called density dependent inhibition. So what that means is if you have, say, like this petri dish, and the cells grow and divide because they're stuck to the bottom, so they're anchored to something, so they're getting that signal. And once they fill up that surface, they will stop growing. And they'll recognize that they have filled up the surface of the bottom, and then they'll be done. And all those signaling molecules will go away and they will not continue on into mitosis. But in this case, if they scrape a few of those cells out, those neighboring cells around that open spot will recognize that they have an open surface. Signal will come in, they can go through replication until they fill that spot again. So those are some things that are part of normal healthy cells: anchorage dependence and density dependent inhibition. In cancer cells, what they do is, for whatever reason, a mutation, either a random mutation or acquired mutation from being exposed to carcinogens or chemicals or radiation, those things can damage some of these control mechanisms. So if you lose your anchorage independence, you might be a cancer cell that can grow out of control in the bloodstream and not have to hook to something to be able to continue. Or if you've lost your density dependent inhibition, you can start growing on top of each other out of control. This is what allows for a tumor to form, right? So like a skin cancer or liver or lung or whatever the tissue is, normally they wouldn't grow on top of each other. That would stop some of those cell cycle signals. But in cancer cells, they've lost that regulation. And so here they can grow on top of each other, and that's what produces a tumor. Now, when we take a look at cancer cells, so here's an example of a tumor growing in breast tissue. So this could be, again, it could be a random mutation, it could be an inherited gene mutation, it could be exposure to carcinogens, something that could trigger a mutation in any one of those regulatory genes. And the cells of the breast start growing out of control, and they're growing on top of each other. They're invading other tissues. They've lost that control, and so they're just going around that cell cycle like they're just out of control. Now, sometimes if you catch that early enough, you can find that tumor, they can go in and they can remove it, a lumpectomy. And if they can get all the cancer cells out, then you'd be cured, and you just have to do some extra checking. If, however, sometimes those cancer cells break away from that primary tumor, they can actually enter either your blood vessels or your lymph vessels and carry those cancer cells to other places in the body. And this is what we call metastasis or metastatic tumors, is they will hop on these fluid freeways like your blood or your lymph. They leave and land somewhere else. So somebody who might have metastatic breast cancer, the tumor starts in the breast, but then it leaves the breast and goes to the lungs or the brain or the bones or the liver or the stomach or the large intestine or wherever. It pops out of those freeways, the blood or the lymph, and starts growing as breast tissue in the liver, in the lungs, in the bone, in the colon, wherever it happens to be. So now you have multiple tumors that kind of broke off from that primary tumor that was in the breast. All right, so this is not explicitly talked about in your textbook, but I really find it important for biology students to know the basis of cancer because it's such a prevalent part of our lives, especially if you're working in healthcare. You yourself may have experienced this, or somebody in your family may have experienced cancer, and the general public doesn't really know too much about the details of what's going on. Now, this is very basic level, but at least now you understand cancer is a result of unregulated mitosis. So what a better place to talk about this than in our chapter talking about mitosis. All right, I will see you next time when we start talking about meiosis. Bye.